Good afternoon, everyone. So it's a special pleasure to introduce an old friend, Dr. Ravi Majeti from Stanford. It's, of course, customary to get up here and say nice things about speakers, but I really mean it when I say Ravi. Ravi is extraordinary at many levels, and so I'm going to take a little bit longer than I usually do just because he deserves it. So Ravi did his undergraduate work at Harvard, then went to UCSF for his graduate work and his postdoctoral fellowship, went back to the Brigham for residency training, and then came back to Stanford in 2004, where he's been ever since. So Ravi was a fellow with Irv Weissman, who many of you know is one of the preeminent stem cell biologists in the world. And during that time, not only really strongly distinguished himself with sort of multiple high-profile papers. I've had to write these down because there's so many, it's actually hard to keep track of. So I should say, and I got ahead of myself, as a PhD student, he had a first author paper in Science and a first author paper in Cell. As a fellow, he had first author papers in Cell, PNAS, and Cell Stem Cell. So obviously incredibly talented as a trainee, and you think, well, maybe he just had good mentors. You know, maybe once he gets on his own, he's not going to do that well, but that's definitely not the case. And I'm just going to give some quick numbers, and I'm just hitting the highlights. In the 9 years since he's been an assistant professor, 2009 till now, 2 papers in Nature Medicine, 2 in Nature Communications, Nature Genetics, Nature Protocols, Cell, 2 in Cell Stem Cell, 1 in Science Translational Medicine, and Cancer Discovery, and those are just the high-profile papers. So I'm actually not aware, I can't think of a cancer scientist anywhere who has that level of productivity in any phase of his career, much less the early phases of his career. So obviously, extraordinary productivity. More importantly, extraordinarily interesting work. So as a fellow, right as he was finishing his fellowship and then transitioning to his assistant professor position at Stanford, of course, Ravi published some of the seminal work that I would, at a fundamental level, put on a par with the CAR-T world. He showed that the innate immune system is extraordinarily powerful, at least in preclinical models, for destroying cancer cells, not only in leukemia and hemolignancies, but also he collaborated with others to also show this in solid tumors. So you're familiar with CD47 or the don't eat me signal. The pioneering work around that was done by Ravi and his colleagues, again, as a fellow and then transitioning to his independent position. So for a lot of people, that would be a career right there. Just developing that, there's a million directions to run. There are now, at this point, Ravi told me yesterday, there's now 6 clinical trials currently in progress to evaluate targeting that axis for therapeutic indications. But what's really amazing is that was just the beginning for Ravi. In the subsequent years, there's no fewer than 5 major areas of investigation that he's pursued. And just to quickly give you a sense of that, he's done seminal work looking at the evolution of how a leukemia stem cell evolves in terms of clonal evolution and the mutational spectrum that leads to leukemia. He's published fundamental work on the metabolism of leukemia stem cells and some of the critical factors that leukemia stem cells are sensitive to. He's published on leukemia genetics and epigenetics at a fundamental level. He published an entirely novel xenograft model that uses miracle ossicle formation to study normal leukemic stem cells. And then he's very recently published a study or the first development of true AML or leukemia-derived iPS cells. So needless to say, not only extraordinarily productive, but extraordinarily broad and impactful in the work he's done thus far. It's incredibly fun to have him here today. Ravi, thank you so much for being here.
Well, thank you, Craig. That's an introduction my mother would be so happy with. It's my privilege and honor to be with you all today, and great to connect with old friends like Craig. I'll just tell you that when I met Craig in 2007, I was a fellow in Irv's lab thinking, how on earth am I going to make a contribution to this field? And at various time points over my training and junior life as a faculty member, Craig has been a good friend and mentor, so thank you to Craig for that. Well, today I'd like to tell you a number of stories about stem cells and AML, and I'll just tell you that our work covers a broad spectrum of topics, all related to stem cell biology, but going into a variety of different areas. And our focus, as you know, is related to this disease AML, and it's one that you're probably very familiar with here with the outstanding group you have focused on this disease. But as you know, it's an aggressive cancer of blood-forming progenitors in the bone marrow, more common in adults than kids, getting more common every year because it's a disease of the elderly. Patients do poorly, although with Dan Pollier's clinical interventions, maybe patients aren't going to be doing so as poorly as the statistics that are published tell us, where patients have an overall survival of about 30 to 40%, maybe lower in the older individuals for whom they're not candidates for aggressive therapy, and the biology of their disease is quite distinct. Now, a lot was learned about the molecular genetics of AML in the era preceding genome sequencing, and some of those points are highlighted here. The key points are that it's a very heterogeneous disorder. There are multiple subtypes defined by either karyotypic abnormalities or molecular mutations. Mutations in the FLT3 tyrosine kinase are very common and are associated with an increased risk of relapse and poor outcomes, whereas mutations in genes like nucleophosmin NPM1 are associated with a better survival. And this is an older plot from the ECOG sequencing data. It's a Circos plot showing a variety of different genes that can be recurrently mutated in AML and how they co-occur in pairs. And I like this plot because it shows you graphically easily that there are lots of combinations of mutations. There's many ways that mutations co-occur to cause AML. So it's a complex disease that has a variety of genetic underpinnings. Now, genetics is not the only aspect of AML biology that we study and that has been studied. And this builds off the work of my mentor, Irv Weissman, and of course many others, with normal hematopoiesis shown here on the left, which is organized as a cellular hierarchy with self-renewing hematopoietic stem cells giving rise to intermediate and lineage-restricted progenitors, and eventually all the mature cells of the blood. Now, John Dick and colleagues really pioneered the work investigating and isolating leukemia stem cells in this disease, and they showed that analogous to normal hematopoiesis, there can be a subset of leukemia cells, the leukemia stem cells, that self-renew and give rise to an aborted differentiation program, but with populations of cells that lack that self-renewal and engraftment potential. And there's immunophenotypes that can be used, or at least in the early days of these experiments, used to identify leukemia stem cells, CD34 positive, CD38 negative, which is analogous to what is known for normal hematopoietic stem cells in humans. More recent studies have added complexity to the understanding of this hierarchy by demonstrating that multiple populations can have leukemia stem cell or engraftment potential, and that maybe these one-way arrows, like we've drawn in this schematic, are not necessarily one way. They could be bidirectional. There can be complex relationships among the different populations in AML. But the fundamental observation that there's a subset of cells capable of initiating the disease in immunodeficient animals has held true throughout all these studies and certainly forms the basis of thinking about leukemia stem cells and their particular properties. So the first story I'll tell you about is one that we recently published, where we were trying to establish better models for xenografting human cells into mice. And this is a bone marrow ossicle model that was developed by a very talented MD PhD fellow in my group, Andy Reinisch. And Andy's now returned back to his home country of Austria, where he's established his own research group. Now, there's a long history of the development of humanized mouse models, and there have been several key time points that led to advances in the field. Really, the early days, they were the SCID-HU mice that were humanized with fetal human tissues that allowed the initial engraftment of human hematopoietic cells. And I would say in the mid-90s, the development of the NOD-SCID strain was a big advance, and certainly Professor Dick and colleagues utilized that strain to lead to some fundamental observations about normal and malignant hematopoiesis. In the mid-2000s, about the time I entered this field, the NSG mouse was bred, and this has really been the revolution for xenograft studies. And there have been ultimately several other different strains that have been developed that have a variety of different immunodeficiencies to permit engraftment and dissemination of a wide variety of human hematopoietic cells. But there are limitations of these available xenotransplantation models, and I also like to refer to this as the dirty little secrets of AML xenografting. First, only about half of AML samples ever engraft in these immunodeficient mice, and this is skewed towards the more aggressive, worse prognosis disease. So we don't really know much about the behavior and the biology of the other 50% of the cases. Secondly, engraftment has historically been defined as a myeloid-restricted engraftment, CD33 positive, in greater than 0.1% chimerism in the bone marrow. Now, you can detect those cells by flow cytometry, you can transplant them from one animal to another, but certainly the clinicians in the room know that that's not leukemia, that's not what patients have, and certainly isn't causing disease in the way that leukemia does in humans. And even if you let those mice go out for a long time. Many engrafting AMLs will never achieve a bone marrow chimerism greater than 20%, which is what we use to make that diagnosis clinically. And there have been sequencing studies, primarily from Tim Lay and Wash Yu colleagues, that determined that in most engrafting mice, only a single clone engrafts, and this could vary from mouse to mouse and even from the same patient sample. So presumably, this is related to the bone marrow microenvironment, and these engraftment deficits can be due to a lack of cross-reactivity between human cytokines, chemokines, and their various receptors, or lack of human cells in the matrix. And as you know, the bone marrow microenvironment is a complex structure. It includes both cellular and acellular components, endosteal, osteoblastic lineages, and a variety of other interactions that are key to the self-renewal and proliferation and quiescence of those stem cells. So we set out to try to generate a better humanized system, and our strategy is illustrated here. We started with a population of human bone marrow-derived mesenchymal stromal cells. These are from a normal bone marrow donor. And we expand them ex vivo under particular conditions, then mix them with extracellular matrix and transplant them subcutaneously into a mouse. Now we can transplant 4 plugs per mouse, right, left, front, and back. And over the course of about 8 to 10 weeks, those cells will undergo differentiation first to cartilage, and then they'll undergo the very important process of endochondral ossification, where cartilage is converted to bone. And it's in that process that bone marrow is formed. So if you take a look at those at the end point, after the 8 to 10 weeks, you can explant them. These are little ossicles, bone structures. They're translucent because the cortical bone is very thin. And in the histology here, this thin cortical bone is on the pink rim, and these purplish cells in the interval areas represent the hematopoietic cells, the mouse hematopoietic cells. So to use this as a xenograft host, we irradiate to condition these animals, and they could be transplanted either by direct transcutaneous intraossical injection because it's in the subcutaneous space and the bone is very thin, it can be punctured, or just by conventional intravenous injection where the cells can hone to those bone ossicles in addition to the native mouse bone marrow. And then engraftment can be followed over time, either by serial aspiration or analysis of these ossicles by histological methods or FACS at the time when the mice are sacrificed. So we wanted to see whether this was really any better than the conventional model, and we took a couple of cases, one shown here, where we knew that it engrafted very poorly into conventional NSG mice, and that's shown here. This is intravenously transplanted into a conventional NSG mouse, no ossicles, and at 8-week time point, we couldn't really detect anything in the mouse bone marrow. And when we ultimately sacrificed this animal out 18, 24 weeks later, there's 0.5%. There's a few dots there if you can make them out, but it's 0.5%. So this falls into the category of engraftment, but that is certainly not recapitulating many of the key features of the disease. So we took that case and we injected it into the ossicles. And the data I'll show you on the next few slides includes both animals that were directly injected into an ossicle, and it would be 1 out of those 4, and the 1 directly injected is labeled in red, and the others on the same animal are labeled in blue, versus animals that were intravenously transplanted where all the ossicles are blue because they were not directly injected. And here you can see with the same case, by 8 weeks, there's fulminant leukemia in those ossicles, starting to spread into the other ossicles. By 12 to 13 weeks, it has spread, and at the end of this experiment, 18 to 24 weeks later, not only has it spread to all the ossicles, it's even established a significant burden of disease now in the native mouse bone marrow. And similar results are seen with the intravenous method, although the kinetics are slightly delayed, but you end up having higher engraftment in the native mouse bone marrow in addition to the ossicles. And presumably this higher engraftment is due to the fact that the bone marrow is continuously being seeded by circulating cells leaving the ossicles, and thereby having much more chance to establish engraftment in those in the mouse bone marrow. This is a summary of data from 4 such specimens, all of which poorly engraft in conventional NSG mice. They all engraft to nearly 100% in the directly transplanted ossicles and quite quickly, which spreads to the non-transplanted ossicles. And in the intravenous mice, it's analogous to what I just showed you. Now, what does this look like histologically? Well, here on the left is an H&E stain, the white outline illustrating the bone here in the pink, with the marrow cavity in between with hematopoietic elements. And this was histology taken from a case in which we had transduced the MSCs used to make the ossicle with a GFP encoding lentivirus. And you can see the GFP positive cells in the bone matrix itself, but there are also GFP positive cells within the bone marrow parenchyma, illustrating that several of the key stromal elements are human derived. I will say that the endothelial cells, which seem to be a very important part of the niche, at least for mouse HSCs, remain mouse in this model. They are not human. Now, when you look here at the engraftment of the AML itself, here's immunohistochemistry looking at human CD45 expression in brown here, and in ossicles that are fulminantly engrafted, you just see a giant collection of those human cells, as you'd expect based on the flow. But in this case, this is an animal that had a directly transplanted ossicle and one non-transplanted ossicle that was lowly engrafted. What you find is that the human cells in this particular case are right up along the endosteal surface. And this is consistent with other data suggesting the endosteal surface provides an important niche for AML. And this is certainly an area of great interest in ours, trying to understand what are some of the key interactions, molecular interactions, that occur at this particular site. Now, I mentioned that AML often contains subclones, and some of those subclones will engraft in some mice and not others, and we wanted to look in these handful of cases whether we were recapitulating the clonal structures of the original patient specimen. Now, this data is very granular, and I'll just summarize it very quickly. What you see on the y-axis are variant allele frequencies, so just the sequencing frequencies of the individual mutations that are listed here. The details of those are not so important, other than to say that we've compared the black diamond here, which is the original blast taken from the patient specimen, to a variety of the ossicle or mouse bone marrow tissues illustrated here. And what you see is we see the same VAF across all of those different locations, suggesting that we're recapitulating the subclonal structure of the original patient sample, which is important when you think about maneuvers to try to develop preclinical models for therapy or other methods to explore how the subclones interact with each other. I will say in one case, we had a canonical NPM1C mutation that was present here at 50% in the original patient sample. This is a heterozygous mutation. And that when we looked in several of the ossicles, this clone was growing, but it didn't expand in the mouse bone marrow itself. So suggesting there may be a preferential growth of those types of clones in the ossicle marrow compared to the mouse bone marrow. Now, I wouldn't be a card-carrying stem cell scientist if I didn't show a limiting dilution assay. And this is a limiting dilution assay to determine the leukemia-initiating cell frequency. So what we've compared here is direct intraossical injection of a native NSG mouse directly into the femoral cavity compared to intraossical injection. You do a limiting dilution across a number of cell concentrations, and you look for the engraftment. In this particular case, the estimated leukemia-initiating cell frequency was improved by about 20-fold in the ossicle compared to the intrafemoral cavity. I will say I was really surprised. In 1 out of 24 cases, we were able to establish leukemia from a single cell, which for me is a holy grail of a lot of our studies to try to get to single cell analysis. This is a second case where the leukemia initiating cell frequency is lower in the thousands in conventional experiment, but it's also improved by about an order of magnitude when looking in the ossicle. So the ossicle indicates an increased leukemia-initiating cell frequency, highlighting the fact that the model being used to explore leukemia stem cells will have a big impact on both the frequency and the particular properties of those cells. And our current work is now focused on 2 primary areas. Number 1 is to really get at single-cell engraftments, both of normal and leukemic hematopoiesis, and also to go back and genetically modify the MSCs to knock out various factors that we think may be critical for interactions with either normal or leukemic stem cells, and be able to analyze that in a human-to-human setting rather than mouse genomics. So now I'll turn to a second area of our genomic studies, and these are a little bit older studies, but they're very illustrative for the way that we're thinking about our biology. And these are studies where we've explored the clonal evolution of pre-leukemic stem cells in AML, work done by very talented grad students, Ryan Corses and Max Jan in my group, along with the postdoc from Steve Quake's lab, Tom Snyder. Now, this is another set of data taken from the genome sequencing of AML. This is the TCGA data that was published in the New England Journal now 5 years ago. And this shows that on average, when they analyzed about 200 cases of AML, that there are 5 coding mutations in likely driver genes in each case. And most cases consisted of multiple subclones, as we've already discussed, based on their variant allele frequencies. Now, we showed that there were pre-leukemic mutations that could be identified in the residual stem cells at the time of diagnosis. And that was really facilitated by our ability to separate out the residual stem cells and genotype them. So this is some of the data that we ultimately went back to do single-cell level analysis and look at the order in which mutations were acquired. And we could find single clones of HSC in this particular case that had a DNMT3A mutation and ultimately track the order in which these mutations were acquired. And it led us to propose a model in which normal stem cells can acquire founder mutations in genes like DNMT3A, TET2, that can regulate the epigenome, and these occur in functional HSCs. Whereas progressor mutations generally occurring in proliferative mutations or proliferative pathways, FLT3 kinase, RAS pathways. These occur outside the stem cell compartment, probably in progenitors, although that is not definitively established, and that ultimately leads to the development of frank leukemia. So this model has a number of implications when you think about how we treat our AML patients. If you consider the time of presentation at diagnosis, there's a population of HSC, some of which are normal, some of which are pre-leukemic meaning harboring some of the mutations. And the leukemia itself can contain many different clones that are, of course, clonally related as well, with these indicated by these colors. We treat our patients with intensive chemotherapy, 3+7 or related agents, and many of them will go into remission, most of them, in fact, but most patients will relapse. And we were trying to explore what could the models for relapse be. Is it that the dominant clone is not eradicated and it comes back at relapse? Does a minor clone grow out? Maybe it has chemo-resistant properties. Do new mutations occur on the background of a pre-existing clone? Or do new mutations occur on the background of a pre-leukemic clone? And now you have a distinct but related relapse. So we set out to explore whether these models could be found by looking at paired specimens of patients obtained at diagnosis and at relapse, and in some cases we even had their remission sample. In fact, when we looked at the remission sample, and all of the data on the next few slides I'll show you is variant allele frequencies on the y-axis with individual mutations on the x, and those that were pre-leukemic at diagnosis are highlighted in blue. What you see is that in these particular cases, at remission, we could identify all the pre-leukemic mutations still in the patient, meaning the pre-leukemic stem cells were not eradicated. They still persisted. Now we looked at the paired diagnosis and relapse specimens, and here is a case where there's a conservation of diagnosis and relapse, between diagnosis and relapse of all the mutations. So this is that option A, the dominant clone at diagnosis came right back at relapse. Here's a case where there are conserved mutations between diagnosis and relapse. But here's an interesting FLT3 kinase mutation that's present at diagnosis in red here, couldn't be detected at relapse. Instead, what we find is a RAS mutation in relapse. And when we went back to deeply sequence the diagnostic specimen, we could find it at a low frequency. So this is a minor clone, the RAS mutant clone, giving rise to the relapse. That's the option B that I described. Finally, here's a case where there are conserved mutations between diagnosis and relapse. And here's a RAS pathway, PTPN11 mutation that's present at diagnosis. It's absent at relapse. And instead at relapse, a WT1 mutation comes up, which we could not detect at diagnosis. So of course, we're limited by the threshold of detection of these sequencing methodologies. But if we presume this is a new mutation, this is either option C or D, where a new mutation has occurred for further clonal evolution. And so even in this small handful of cases, and I'll just say that we have a much larger cohort that we're actively analyzing the genomics from now, where we hope to be able to elucidate the relative frequencies of these different types of models and whether or not more than one of these mechanisms is in play in any individual patient. But if you think about the cartoon here, what we're looking for and hoping for is that we'll end up with patients where we can eradicate both their red leukemia clones, their colored pre-leukemic clones, and they'll get long-term disease-free remissions. And ultimately, we don't want to end up in a scenario where we either have minimal residual disease with persistent leukemia cells or fulminant leukemia developing on the background of pre-existing pre-leukemic clones. Now, through the course of all of these studies, we looked at the pre-leukemic stem cell compartment in about 40 cases. It was 39 cases here. And we define a metric we call the HSC pre-leukemic burden. So this is a little complicated, but the stem cells at diagnosis, not the leukemia, but the leftover stem cells, they will range in their level of mutations, whether they have the initial hits. And so we stratified them according to how high a level of those initial hits they had. And they ranged from basically nothing low burden to a very high burden, virtually 100% of those stem cells. And they're all with the various mutated genes that you see here. And we stratified these into low burden and high burden. And we were really surprised to find that the patients who had a high pre-leukemic stem cell burden did much worse than those that had low. Now remember, all of these people went on to get AML. The genetics of their leukemia is very similar. They were treated very similar. We're just talking about their stem cell compartment at the time they were diagnosed. And so we don't know if these are cell intrinsic effects or cell extrinsic effects, microenvironmental influences, etc. But this is certainly something we're trying to get more N on in order to be able to investigate this issue further. Now, I mentioned that many leukemias are associated with a number of different subclones. And the next story I'll tell you is about pluripotent reprogramming of human AML to try to explore some of the subclone issues or mechanisms involved. And this was work done by an MD PhD student, Mark Chow, in my group, who collaborated with a whole set of folks in the lab to study this. And it comes initially from thinking about the epigenetics of AML. And there are certainly a large number of genes, many of which are highlighted right here, that are mutated in AML, and certainly a number of epigenetic therapies that are either approved in the case of the hypomethylating agents and now the IDH inhibitors, or under investigation in AML. And one question we were approaching is, what's the relative contribution of epigenetic compared to genetic abnormalities to the disease of AML? And we decided to take a strategy to try to generate cancer-derived iPS cells to both reset the epigenome and see whether or not the genetics could drive the disease back, and also to be able to understand aspects of subclonal structure. So you're all, of course, familiar with iPS cells and the pioneering work of Yamanaka to develop those cells. But in the case of cancer, it's always been quite a bit of a challenge to generate cancer-derived iPS cells because of the mutations they harbor, as well as their DNA damage repair defects they may have. So we set out to ask very simple initial questions. Can AML cells be reprogrammed into iPS? Is epigenetic reprogramming sufficient to reverse the leukemic disease? And can we use iPS cells to think about therapeutic targeting? So we started with 2 cases of AML from our tissue bank, both of which had MLL fusions. So these are translocations of the MLL gene, commonly found in infant AML, but also found in a proportion of adult AML. And we chose those for 2 reasons. First of all, the ability to detect the translocation by FISH allows us to track very easily what the clonal origin of the cells that we were studying is. And also because these cells have a tendency to proliferate in vitro at least a little bit, and you need proliferation in order to get iPS cells to form. Now, I'll also point out that both cases had corresponding RAS mutations, which is very common in MLL rearranged AML. And so the strategy was to take the primary patient blasts, and we used a non-integrating Sendai virus approach to introduce the Yamanaka factors and then derived AML iPS cells. And without showing you, I'll tell you that we made iPS clones. They expressed pluripotent markers. They formed teratomas when transplanted in vivo. And we then looked to see whether those clones... Oh, my screen is blank. Okay, sorry. We then looked to see whether those clones had the genetics of the AML, or were they uninterestingly derived from the very rare but residual normal cells. And we looked first by FISH. This is a break-apart probe, so the yellow signal is wild type, the break-apart red and green is mutant. And in the patient specimen, of course, we see that the FISH is showing the MLL rearrangement. And here's one of those iPS clones also showing the MLL rearrangement. Again, this is variant allele frequency sequencing of the particular single nucleotide variants present in this particular case, and the black is the primary patient sample, the colors are the different iPS clones, and you see the same variant allele frequency in all of these clones, illustrating that, yes, these clones are derived from the leukemic clone. All of them except for this one right here, clone number 3, which is wild type for the KRAS mutant, and it was just fortuitous that this happened because it allowed us to study this in more detail. And when I say fortuitous, if you think about it, the black bar here from the patient sample is virtually 50%. You would think that almost every single cell in this patient has this heterozygous KRAS mutation, but this particular clone did not. All right, so we derived these iPS cells, and the first thing we did was we tried to differentiate them into various lineages, and we worked with our colleagues in the Stanford Stem Cell Institute, and to our great surprise, they made completely normal cardiomyocytes, able to handle calcium and generate the appropriate electrophysiological responses of cardiomyocytes. The same was true with their ability to make neurons. They made completely normal neurons that could fire action potentials and had no evidence of dysregulation. But when we differentiated them into blood and we looked at their in vitro colony formation, what we found is all of the AML-derived colonies failed to make erythroid colonies, which the normal controls did. And these colonies could perpetually be serially replated, illustrating either an increased proliferative potential or a self-renewal potential in this particular assay. But not being content with in vitro, we decided to go one step further and transplant these differentiated blood cells from the iPS into the NSG mice. And we found to our great surprise that they would engraft and give rise to a myeloid-restricted leukemia that was present in the hematopoietic organs, formed very large spleens and the morphology of the leukemia. We genotyped those cells, and yes, they had all of the same variants in the original iPS clones derived from that patient sample. And when we transplanted them into secondary recipients, they gave fulminant disease. So the genetics trumped the epigenetics. You could reset the epigenome, and the MLL fusion and other mutations could drive it right back to form leukemia. Very surprising result, and I actually didn't believe it worked until the postdoc showed me the data. Okay, but I mentioned that there's another way to think about the ability to use iPS clones to look at subclones, right? iPS cells are all single cell derived. And we had this particular cases here where we have these KRAS mutations or NRAS mutations that sensitize these cells to MEK inhibition. So we went back to look at MEK inhibitor activity against these various subclones. And I come back to this case 42 here, where we know at diagnosis the vast majority of the cells are KRAS mutant. That's the red colored clone here. But there is a very rare but minor clone that's KRAS wild type. So we wanted to see whether we could explore the sensitivities to MEK inhibition in vitro. And we found, as you might predict, that the KRAS wild type clone was insensitive to MEK inhibition, whereas these two KRAS mutant clones were sensitive to MEK inhibition, and that's shown here with an IC50 curve illustrating the same thing. Now, in this particular case, this patient had been treated with a anthracycline, cytarabine induction chemotherapy, and fortunate for us who are interested in studying this, but not fortunate for the patient, they relapsed. And we were able to collect their relapse specimen, and when we genotyped that relapse specimen, which is shown here in the red dot, we found that it was KRAS wild type. Surprise, surprise. So it's this very minor KRAS wild type clone. It acquired some additional mutations in the course of its clonal evolution that ultimately gave rise to its relapse. So we went back to the iPS clones and looked at their sensitivity to cytarabine, and we found that the KRAS wild type clone was less sensitive to cytarabine than the KRAS mutants, which is a little counterintuitive that you would generally think that the more mutated the cell is, then the more chemo-resistant it's going to be. And that was true here with the IC50 curve as well. And we finally went back to our tissue bank, collected a bunch of other patient specimens that were known to be MLL rearranged and genotyped them for their RAS pathway mutants. And we found that in this collection of specimens, the KRAS wild types all showed less cytarabine sensitivity than the KRAS mutants. So here we've taken individual patients, reprogrammed the iPS, make these individual subclones, and found a principle of therapeutic sensitivity based on those studies. I don't suggest that making iPS cells is something that one would do in a clinical scenario for therapeutics discovery, but there are certain scenarios where it can be very useful, like the one I described here. And certainly the concept of being able to physically separate subclones by using an iPS approach is one that could allow you to study their different properties. So now I'll move on to a little bit more recent work. where we've been carrying out genome editing in human HSPCs. And this is all part of our technology building to be able to do as many manipulations as possible in primary human cells. And this is work of a couple postdocs from my group, Andy Reinisch and Rajiv Sharma, with two postdocs, Rasmus and Danny, from Matt Porteous' lab at Stanford. And many of you may know that Matt is a real pioneer in genome editing in human HSPCs, and we're very fortunate that our labs are just across the corridor from each other. So everybody knows about the CRISPR-Cas9 system, but what I want to illustrate are how we utilize this in primary human cells. So we use the Cas9 and guide RNA as a pre-complexed ribonucleoprotein that we transfect into primary HSPCs, and this site specifically introduces a double-stranded DNA break. And this can be repaired in two different ways. The first is through non-homologous end joining, which is error-prone, as you know, and can introduce small insertions or deletions that can ultimately result in frame shifts and premature stop codons. And this is a very important way to knock out genes effectively. But an alternative way to repair the double-stranded DNA break is to provide a template with regions of homology that flank the site of the double-stranded DNA break, and can carry a payload that can get recombined into the locus of interest. And we do this by transducing those cells with a recombinant AAV6 vector that we've engineered to have what we want. And so the result is that homology-directed repair allows disruption and modification of the desired genomic locus. And the inclusion of a fluorescent reporter cassette, so that what could be included in between the arms of homology, could permit cell isolation and tracking, which is necessary for the downstream applications in the research applications. So first of all, does this work? Well, this is some data showing HDR, homology-directed repair, mediated gene editing. And on the y-axis will be a reporter. In some experiments, it's a GFP, BFP, RFP. You get it. It's various fluorescent reporters. And here, these are mock cells. Of course, you see no cells that are fluorescently labeled. And if you just transduce with the AAV, no Cas9, no guide, no double-stranded DNA break, the AAV will transduce those cells, and you can see some expression. But when you put it all together with the Cas9, now you see a large population of fluorescent labeled cells. These can be purified. This is data editing the CCR5 locus, and you see that those high fluorescent expressing cells stay fluorescent throughout their time in culture. Some of these intermediate cells are episomal, and they clearly lose their signal, but some integrate, and you do get the signal. And it's not just random integration. You can design inside-out PCRs with primers that interrogate the endogenous locus as well as the modified locus, and we find what you'd expect. In some cases, they're biallelically modified loci, in others, they're only monoallelically modified. This is shown here for the CCR5 locus. This is data showing that with a couple of different loci, you can get genome editing with either monoallelic or biallelic integrations. Now, what happens in this case when you use one color and you see that you might have monoallelic recombined loci is you got to look at the other allele to see what happened there. And what happened there is what you would expect. In some cases, it's just wild type and it hasn't been edited. In other cases, there's an indel that's in frame, but in most cases, it's an indel that's out of frame. And that's true across the different loci that I've shown you here. So how can we use this to start exploring the behavior of HSCs and HSPCs? Well, we started with a disruption of the STAG2 gene. STAG2 is a member of the cohesin complex, and we and others had previously shown that when you knock out cohesin in HSPCs, there's an increase in self-renewal, a decrease in a decrease in erythroid lineage differentiation. And we wanted to see if we could recapitulate that. And the other advantage of utilizing STAG2 for these studies is that it's on the X chromosome, and so we're using our cord blood units that we procured from males so that we know that we've got the genotype that we want. So we carried out our editing at the STAG2 locus and placed those cells into colony-forming assays, and as we anticipated, they don't form any erythroid colonies, but they form plenty of other colonies, illustrating this process hasn't somehow caused these progenitor cells to lose their progenitor function. And in fact, when we cultured these cells further in vitro, again, we're looking at the GFP-positive edited cells, and the cohesin-modified cells show a huge striking retention of CD34-positive cells in culture compared to control. And I'll just mention that in the controls, what I'll show you is we use the AAVS1 locus. This is a relatively a safe harbor in the human genome, analogous to ROSA 26 in the mouse, if people are familiar with that. And so the cells are edited at AAVS1 in the identical way. Now, here's an example where we've gone in vivo with this. We start with cord blood cells. We're using now an editing strategy in the RUNX1 locus that knocks out the RUNX1C isoform. And we transplanted those cells in vivo, and they do engraft. They give rise to bilineage lympho-myeloid hematopoiesis, and they're all GFP-positive cells. So the cells still have their HSPC functions, and if you leave these beyond the initial 8 weeks, out another 8 weeks, the engraftment expands, and the cells remain with their differentiation ability and their fluorescent tagging. Not to be outdone, the guys in the lab decided to go all out, and then they decided that instead of adding one virus that had a GFP tag, they'd add two, one GFP, one BFP. And of course, the idea is that if you have double positive cells, now you know exactly their genotype. They are complete knockouts at both alleles. And yes, this works. Here you can see about a 10% efficiency. This is in the ASXL1 locus, and these persist. And when you do the genotyping by the PCR, it's as you'd expect, that you've integrated into the endogenous locus. And then the guys went really crazy, and they decided to add multiple colors. So now they've got green and blue in the RUNX1 locus. They've got red and crimson in the ASXL1 locus. Can they make completely quad allelic knockouts? And in fact, at 1.1% of the cells, which frankly is enough to do experiments, they do develop cells that have that potential. And in fact, they still are able to form hematopoietic progenitor colonies in vitro, so they haven't somehow lost their function. So using these strategies, we've targeted a number of different loci of interest for leukemia and HSC biology. Of course, the frequency for single allele and single gene targeting is much higher than with multiple different multiplexing, but still we're able to accomplish this across a wide range of different strategies. Now, knocking out genes is great, but a lot of what goes on in hematopoietic malignancies and AML are point mutations, or heterozygous mutations. And so you could imagine strategies to make heterozygous knockouts where you integrate a wild-type cDNA along with the tag in one allele as opposed to just having the knockout in the other. Or you could make SNPs using the same thing where you have one wild-type cDNA, one SNP cDNA. So those are strategies that can be implemented. But what I'll show you is an alternative strategy to engineer a point mutation into these human HSPCs. And this is where we've engineered the canonical DNMT3A R882H point mutation by using a splice capture strategy. So this is the endogenous DNMT3A locus. R882 is located in exon 21. We make a double-stranded break in the intron upstream of that, and into which we integrate an engineered element that has a splice acceptor and the remainder of the DNMT3A cDNA, either wild type or mutant. So now, expression coming from the native promoter will splice onto this engineered element, make the rest of the cDNA. And here, instead of a fluorescent tag, we've used a truncated NGFR receptor tag, which can be antibody stained on hematopoietic cells. And I wouldn't show it to you if it didn't work. It does work. Here we've got about 20% NGFR positive cells that go into immunodeficient animals. They engraft very robustly with both lymphoid and myeloid hematopoiesis and still retain a significant population of NGFR positive cells. And because Sanger traces are boring to look at, you can believe me, I hope, when I tell you that we isolated these cells, Sanger sequenced their cDNA, and they have the R882H mutation in the cDNA. So you could use different combinations of wild type and mutant variants to make either homozygous or heterozygous variants of the DNMT3A. And I'll just say that we've now, using this strategy, made a variety of other point mutations like NPM1C, the IDH1 and 2 canonical mutations, and different team members are now studying their various biology, starting to multiplex them together to allow us to explore how different combinations of mutations can interact. So I'll say that this CRISPR technology has exploded in the lab with the postdocs taking it to its limits. All right, maybe in the last set of studies I'll talk about is some more therapeutically oriented studies that involves the synthetic lethality as it relates to IDH mutations in AML. And this is work that a postdoc in my lab, Dan Thomas, along with a computational biologist from the Stanford Computer Science Department, Subarna Sinha, have done, and really builds off of work that Steve Chan did in my lab before he left to start his own group in Toronto. And synthetic lethality is this concept that there can be gene or pathway, let's say A or B, when either one of them is disrupted, cells survive just fine, but when both are disrupted, either by mutation or genetic means or pharmacologic means, this results in cell death. And a lot of large-scale omic-level studies in cell lines and others are implemented to try to identify synthetic lethal relationships, particularly for undruggable cancer mutations. But there are limitations of large-scale cell line screens in particular. There can be off-target effects, particularly with shRNAs, but to a lesser extent with CRISPR, and sometimes incomplete knockdown. There can be an inadequate number of cell lines. Particularly, there can be inadequate representation of important mutations. For example, we have a panel of about 30 AML cell lines that we use extensively in our studies, that we've genotyped and obtained them from ATCC, and there are no native cell lines that have endogenous IDH1 or IDH2 mutations, and there are few with some of these other common mutations. And of course, cell line screens may not ultimately reflect what goes on in the in vivo leukemia microenvironment. So our talented computer science colleagues developed an algorithm called MISSILE, which is illustrated here. And this is a purely computational method, no experimental data involved, to try to identify candidate synthetic lethal interactions. And so this takes advantage of the fact that many mutations in AML in particular are present across other cancers, and utilizing the TCGA pan-cancer genomics data. So you start with a mutation, you look for the cancer type of interest, and you select the other cancers that also have that mutation. And now here's the real key. You're asking the question, are there more copies of the gene in the mutated samples? And it's more easily illustrated by what's shown here on the right. These are all Boolean analysis and Boolean implications, but you want to identify a case where whenever a gene is mutated, IDH1, a particular other gene is never deleted. Right? This might suggest a synthetic lethal relationship because otherwise those cells have dropped out. The alternative is that whenever a gene is mutated, that second gene is amplified, and it's never amplified unless that mutation is present. So this is, again, the same idea about the copy number. And then we apply some expression filters to make sure the genes are going up or down in the right direction based on that, and out comes a list of synthetic lethal candidates. So again, it's a purely computational framework taking primary human data, not cell lines, to try to identify candidate synthetic lethals. So first of all, we wanted to know whether MISSILE actually works, and so we took some data from the Achilles project out of the Broad, where they had done large-scale shRNA screens. This is data from colorectal cancer cell lines, where they had enough cell lines to derive some statistical power. And using GSEA as the statistical test, we found that MISSILE did enrich for synthetic lethal genes that had been identified in these screens. Now, we had done our own screen. Steve Chan had done this in the group, looking at an IDH1 mutant cell line. In fact, this is a cell line we engineered to be inducibly express mutant IDH1. So we have a variant of it that has wild-type inducible expression and a variant that has the mutant inducible expression. And we carried out a synthetic lethal screen here. And so we wanted to see whether MISSILE overlapped with the results from our screen. So we have our own screen here and all of its outputs. We have the MISSILE candidates, and we applied a statistical Fisher's test, and there was a strong enrichment of those that MISSILE predicted that had come out in the screen. In fact, the most strongly identified from the screen, which was a depletion screen, were ones that had been predicted by MISSILE. So we found that the BCL2L2, this is one of the BCL2 family members, came out of the MISSILE screen. And in fact, the original study that Steve conducted had shown a synthetic lethal relationship between IDH mutations and the BCL2 family members. But the question was, could MISSILE identify any new targets for IDH1? So MISSILE produced 89 candidates computationally predicted from the algorithm. And we took those 89 and put them through a databases for those that were druggable, and out we popped 17, of which we obtained some inhibitors from Sigma and other chemical suppliers, and we tested them against our inducible cell line. And so here we can either give doxycycline to express the mutant or not, and looking at the relative killing effectiveness. And the strongest hits were the BCL2 inhibitors, ABT199 and 737. But our interest was drawn to this other hit called TOFA, T-O-F-A. This is a small molecule inhibitor of the gene called ACACA or ACC1. This is a gene that's called the acetyl-CoA carboxylase, and I'll talk a little bit more about it. But this is what the missile output was. We found that there was a complete mutual exclusivity between the presence of the IDH1 mutation and deletion of ACACA, and the expression went in the way that it was expected to. So we set out to validate that first genetically. We used shRNAs against ACACA. We're using that THP1 cell line that's DOX-inducible. And when we turn on the IDH1 mutant in the presence of those shRNAs, the cells drop out. That's true also when we do it with a CRISPR-based system. Here again, when DOX is added in the presence of the mutation and the knockout of ACACA, you see the dropout of those cells in these couple of different methods of applying CRISPR across these cell lines. What about primary AMLs? Well, here we've, well, first of all, this is the pharmacologic experiment on the left. This is showing not the genetic knockdown, but validating that TOFA, this small molecule inhibitor of ACACA, again, those cells drop out when DOX is added to express the mutant. And we saw that there was an increased sensitivity of primary AMLs that have any mutation in IDH1 when exposed to TOFA. Now, TOFA is a poor drug. It can't really be used well in vivo. It's really just a pharmacologic tool. So to validate this further in vivo, we took a primary AML that was IDH1 mutant natively, transduced it with an shRNA encoding lentivirus, shRNA against ACACA, and then looked at its engraftment over time and saw that there was a marked reduction in its engraftment relative to the control. So we think these experiments ultimately validate those findings that ACACA is a candidate synthetically lethal partner for mutant IDH1. But as you know, IDH1 mutation is a target of AG120, the Agios inhibitor now recently approved for the treatment of IDH1 mutant AML. And we wanted to see what was the effect of combining ACACA inhibition with AG120. And in fact, you see there's no reversion of the effect of TOFA. So AG120 does not rescue the effects of ACACA inhibition. That's true in primary cells as well as the cell line. And this is an important point because AG120 will completely block the production of 2-HG, the oncometabolite that the IDH mutants make. And so this effect, the inhibition of ACACA, is 2-HG independent. And that is unlike the BCL2 effect that we had seen before, which we think is 2-HG dependent. So what is actually going on? Why ACACA? What is the metabolic behavior that we're exploring here? Well, IDH1 and IDH2 carry out a number of different biochemical reactions, and I do not purport to be a metabolism expert at all. But one of the functions and one of the key differences between IDH1 and IDH2 is that IDH2 is in the mitochondria, IDH1 is in the cytoplasm. And in addition to its canonical role of converting isocitrate into alpha-ketoglutarate, IDH1 can carry out a reductive carboxylation from a glutamine pathway that produces isocitrate that is in turn shunted to make acetyl-CoA, which ACACA acts upon to make malonyl-CoA, and this is the key rate-limiting step for de novo fatty acid synthesis. So we wanted to understand whether there are metabolic differences between IDH1 and IDH2 mutant AML, and as it relates in particular to fatty acid synthesis. And it turns out that ACACA gene expression is higher in IDH1 mutant but not IDH2 mutant, suggesting that there can be a difference. And when you consider the biochemistry of IDH, 2-HG is not the only perturbation. There's a defect in NADPH production that they both share, but more specifically, only IDH1 is really defective in this reverse reaction, the ability to make isocitrate for lipid synthesis. This is an IDH1 specific effect. So we wanted to ask, does IDH1 mutant AML exhibit defects in lipid metabolism? And we had to develop some mass spec methods to be able to detect lipids. There have not been reliable methods to detect lipids from a reasonably small numbers of cells. Most prior methods require very large numbers of cells. But we worked with a very talented biochemist at Stanford, Gary Peltz, and he developed a variety of methods to be able to measure these things, the details of which I do not know in great depth, but we did validate these approaches by using a variety of standards. And we first applied this lipid metabolic profiling to the IDH1 and IDH2 inducible mutant cell lines, and we found indeed in this PCA plot, looking at the non-polar, the lipid metabolites, that IDH1 was very different from IDH2. And when you drill down into that, IDH1 mutants have a big decrease in production of a variety of these lipid species. And looking at this more from a Venn diagram point of view, polar metabolites, 2-HG and related molecules, there's great overlap between IDH1 and IDH2, but in lipid metabolites, there's not. The IDH1s have this big reduction that is not seen in the IDH2s. Now, we went on from that to do primary sample metabolomics, taking a number of primary AMLs with IDH1 mutants, some with IDH2 mutants, some normal karyotype AMLs, as well as normal progenitors, put them through the same assay. And now this heat map shows that the IDH1s clustered by themselves and the IDH2s clustered more with the normals. And when looking at a volcano plot comparing IDH1s versus IDH2s, all of those that are significantly decreased in the IDH1s are multiple intermediate chain fatty acids, and that's what's illustrated here. Many of those species we were able to molecularly identify. I don't expect you to read this or see that other than to say that the IDH1 showed much reduction here, red, compared to the IDH2 of a variety of different phospholipids. And in fact, what we found, oh boy, computer has frozen. Sorry, everybody. Well, I'll just tell you, what we found was that all of those phospholipids were decreased, and those are the lipids that are involved in making the lipid bilayer. They're critical for cell proliferation. Is it come back up? Okay, good. All right, so in the last pharmacologic experiment we did was to show that TOFA showed a preferential inhibition of the IDH1 mutants compared to the IDH2. Okay. Now, it turns out that IDH1 and IDH2 are mutated at relatively equal frequencies in AML, but that's not true in other cancers, where IDH1 is mutated in a whole variety of brain tumors as well as sarcomas, and IDH2 is not seen to be mutated in those cases. And we wondered whether this metabolic difference might help explain some of that. So we started with we took an IDH1 mutant sarcoma cell line, knocked down ACACA, and found that it didn't grow in vivo. And unlike IDH1 wild-type cell lines that do, we found this was due to a defect in proliferation with Ki67 staining, as shown here, and that when we used AG120 in this same model, we didn't revert the effect. Again, AG120, 2-HG is gone, but you'd still see the benefit of ACACA knockdown. Now, since this is involved in fatty acid synthesis, we put these animals alternatively onto a lipid-free diet. And in fact, we found that a lipid-free diet here in pink made the survival benefit even better. So you blocked ACACA with shRNA, and you add the lipid-free diet, and the effect is even stronger. Now, this is in a cell line of a sarcoma model. And here's a primary AML, IDH1 mutant AML, transplanted into mice, put onto either a normal or lipid-free diet. And in fact, the lipid-free diet animals do better in terms of their burden of leukemia. And so what we come up with is just different ways to target IDH1 mutant cancer. Of course, there's AG120, and ultimately there are several other IDH1 mutant-specific inhibitors in development. ABT199 affecting BCL2. We think this is a 2-HG-dependent effect. But inhibition of ACACA with TOFA is another way to achieve therapeutic efficacy against IDH1 mutant cancers. And I'll just say that we're working with our chemical colleagues to generate better small molecule inhibitors of ACACA in the hopes of being able to do more pharmacologic studies in our in vivo models. So with that, I'll stop. I've tried to acknowledge all the people who've done the work in the group in each of the different projects. And I just acknowledge our patients who've donated their specimens and funding from all of these agencies. So thank you, and I'll take a few questions.
The future is about targeting any cancer at early point possible. So I was thinking about your pre-leukemic work, real interesting, and also thinking about how we could marry that with your genome editing technique. So my question is, would you use your genome editing technique to create some pre-leukemic mutations so you can monitor their response or kinetics in the presence of chemotherapy, or then in this case also to identify drugs that perhaps can be eradicated?
Absolutely. We have ongoing projects. Yeah, the question was really, whether we could use the genome editing to engineer pre-leukemic cells or pre-leukemic stem cells and then study how it is they expand, what their chemosensitivities are, and the relationship to other clones. That's like aim 1 and 2 of my ongoing R01, and we're using those exact methods to do that. I will say it's interesting, if you think about it, it's the same mutations, like the IDH mutations. They can be pre-leukemic. They are the targets of these inhibitors, but do you think that they're going to be sensitive in the same way the leukemia cells? It could be, it could not be. And I think CML is illustrative. The stem cell compartment is insensitive to TKIs, but the progenitor compartment is sensitive. There's no reason to say necessarily that the pre-leukemic stem cells have to show the same sensitivity as the AML cells. We've got to explore that. Trisha.
So way back to the obstacle story, it's not surprising that the leukemia initiating frequency would be higher in that setting. I wonder if you ever had a chance to revisit the identity of the cell itself in that setting versus, the traditional way.
Yeah. It's a great question. The question is whether using the ossicle and seeing an increased leukemia stem cell frequency, well, frequency is one thing. What about the phenotype? What about their properties? We haven't done those fractionation studies yet. I think it's really important. I guess I'm much more driven by the idea of trying to do single cell transplants and be able to actually get at the stem cells by doing single cell transplants. But I take your point. Sometimes my own postdocs complain I ask for things that are too hard to do. Dan.
So back to your sort of origin of relapse work, and we know that well. I think I remember your plenary session on this, but have you been finding differences with respect to the duration of response? Like for instance, that scenario E, where it's almost like lightning striking twice the development of a new leukemia from the same precursor sort of population. Is that like do patients stay in remission longer?
Anecdotally, it looks like that's the case, but we need bigger N to be able to tease that apart. And I think you're right. I think that late relapses may be from patients whose pre-leukemic clones re-evolve.
How common is that scenario? It just seems if that truly is happening, it's just really going to be a big chapter. Almost curing the present disease and then just putting out a new fire. I mean, is that a rare thing to happen?
I think it's relatively rare at this point, but we get better therapies that treat up front that don't eradicate the pre-leukemic compartment, and maybe we're going to end up seeing more late relapses. It's a great question. It's an open one.
All right. Thank you, Ken.
Yeah, thank you, everybody.
Well.